Thank you, Professor Pedro, for your nice talk before. I'm Roberta Teixeira Polez. I'm a postdoctoral researcher at Aalto University. I just recently defended my PhD. So today I will talk a bit about a few results from my 4 years of PhD on the topic of hybrid polysaccharide hydrogels for cell culture platforms. So the focus is on biomedical applications and tissue engineering. So let's see. Yeah, you can see, right? Yes. So biomaterials and the use of biomaterials play a fundamental role in healthcare and biomedical engineering. And when I talk about biomaterials, I talk about materials that have direct contact with cells, and they are usually used to restore, replace, or even enhance the cell function and or cell tissues. So by designing these biomaterials, we can develop better cell platforms for culture applications, and these have potential to improve the medical research in general and reduce the costs of research and future applications, and also enhance the regenerative therapies in the future. So my materials used in biomedical applications are usually animal-derived. So they have similar structure and composition to the human tissue, but at the same time, you have this batch-to-batch variability, which hinders the reproducibility of these materials. It can also have some risk of infection and immunogenic reactions. And also there is always the possibility on the risk of transmitting diseases from animals to humans. The synthetic materials used in this area, on the other hand, are more reproducible, but they are often non-biodegradable, and they can have some rejection reactions, but they are usually inert. And in this sense, the plant-based materials are gaining a bit more interest in this area because they are renewable, they are abundant, they are also biocompatible, and they have low risk of rejection, and there are no infections. So there has been some development in this field. Regarding the ethical issues on the use of animal testing or even animal-based materials for medical research, there is this global trending where the world is trying to replace these animal-based tests when possible for non-animal-based. Also reduce the number of animals to a minimum, and also refine those tests in order to improve the welfare of the animals in this animal studies and animal testing. So here in Europe, there is this directive that aims to replace all animal research for non-animal-based methods. And also recently, last year, the FDA removed the requirements to use animal tests for a few classes of products. So the world is kind of transitioning towards a less animal testing and better practice of science. So this creates opportunity to use to develop new biomaterials based on natural polysaccharides. So in this presentation, I will focus on a few polysaccharides. So I think the most common one is the cellulose, and also chitosan, as also Professor Pedro mentioned a few applications of chitosan, which is derived from crustacean shells by the acetylation, often used as drug delivery platforms and wound healing material. Also xanthan, which is a polysaccharide produced by some bacteria. And this tragacanth gum, which is also a polysaccharide from trees like cellulose, and is exudated by the tree's barks, like this crystal yellow materials. And both xanthan gum and tragacanth gum are usually used for in food industry as thickener and emulsifier agents in some formulations. So these polysaccharides, when they are in water media, they swallow and they form these gels, and they have potential application in biomedical materials. So what I was aiming to do is valorize this kind of polysaccharides towards new applications. But for use these materials in biomedical and tissue engineering, you need to have optimal properties or appropriate properties, as also discussed by Professor Pedro previously. Especially cell compatibility, which is the first. So the material shouldn't be toxic or hinder the cell proliferation or growth. The material also should have these appropriate mechanical properties which match the intended target tissue or application. The porosity should be optimal to accommodate the cells and also allow mass transport and fluid transport, which is also related to swelling. The rheological properties of this material should also direct the processability and the performance of these materials. And the degradation, if it should stay inert inside the body or if it should slowly degrade and be absorbed or be metabolized. So all these properties need to be tuned or should be controlled for different applications. So my aim here was to use these polysaccharides and combine them to improve those properties. So I'll talk about a few examples. So the first one would be the chitosan. So the chitosan has intrinsic antibacterial and antifungal properties. And here we combined chitosan with the fluoroglucinol, which is this phenolic compound extracted by, it's extracted by some plants and produced by some microorganisms. And combining these two materials, we aim to develop some material, some hydrogel for, I'm sorry, I can hear an echo? Okay, sorry. So the aim was to use those two components to prepare hydrogels for wound healing and also local or transdermal drug delivery. So the process to produce them was fairly easy and straightforward. So we dissolved this chitosan in acetic acid and this fluoroglucinol in some mix of alcohol and water. in different concentrations, and we put them in molds, and we gelated them via salt gel transition in a vapor chamber. And after this gelation finish, we rinse them and to remove the excess of the solvent. So we have this chitosan hydrogels prepared, and we freeze dry to have these scaffolds to further characterize. So to understand what's happening in the molecular level, we did some rheology tests. And here you can see the storage and loss modulus as function of strain. For the chitosan, and then we prepared this chitosan with 50%, 50 milligrams per milliliter of fluoroglucinol, and also 200 milligrams per milliliters of fluoroglucinol. So we can see here that the increase in the fluoroglucinol increase significantly the moduli, the storage and loss modulus of these hydrogels. And what we see is the adsorption of the phenolic compound on top of the chitosan chains. So they are interacting, and they are the fluoroglucinol is acting as a physical cross-linker. So we see a bit denser structure and increased modulus. So if we characterize the structure of these hydrogels, of these cryogels after freeze drying, we see the chitosan one has very porous and interconnected structure of pores. And when we added more fluoroglucinol in it, the phenolic compound, you have more a dense and packed structure, as you can see in these SEM micrographs. What we could also see is the effect of the fluoroglucinol on the pore size and also the porosity. So we can see here the chitosan has a big pore, 97 micrometers. And when we increase the fluoroglucinol content, we have diminished to 38 micrometers. So it becomes a denser and stacked and compact structure. And it didn't affect much the porosity overall from 97 to 94. So we wanted to use these hydrogels for a wound healing application. So we need to test the hemocompatibility. So if you put in directly in the wound in contact with blood, it needs to be kind of inert or not interact. So here we see the hemolytic activity of the hydrogels. They were pretty low. So they are considered safe to use because above 5, it's already considered hemolytic. So they're not lysing the blood cells and breaking and damaging the structure. Also, to use in tissue engineering, it needs to be biocompatible. So we tested the proliferation through double ST1 assay for human dermal fibroblasts. For it's not written the days, but it's 1, 3, and 7. So in day 1, we see here that the proliferation or the viability of the cells is around 100. So there was no significant decrease in viability. So it's not considered toxic. And after 7 days, we can see a bit of the decrease, or it's compatible, but the cells proliferating a bit slower than day 5. To assess also the versatility of these hydrogels for drug delivery systems, we incorporated some model drugs. So we chose this ciprofloxacin, which is an antibiotic drug. We incorporated it and checked the cumulative drug release. So after 6 hours here, we see that the chitosan release around 20% of the drug, and for the higher concentration of phenolic compound, we have the increase to 40%. So we were able to double the amount of release of drug just by tuning or mixing or having a physical crosslink and not chemical crosslink in this hydrogel. So we proved this to be an efficient way of not using also toxic crosslink that could harm the cells and hinder the wound healing effect or the proliferation. So moving to a second example. So we use here tempo-oxidized cellulose nanofibrils and combine it with the tragacanth gum, which has a composition of basically the main components: galacturonic acid, galactose, and rhamnose. So thinking about the main processability or the main properties to achieve a good printability, there are a lot of parameters that have a part in it, such as the viscosity and the modulus, the shear thinning, the yield stress, and filament. So all of these parameters should be optimal for you to achieve good extrudability of your gel, and for your also your printed scaffold not to collapse. So here we optimize the rheological properties, and we printed the scaffolds. And here we have presenting only two of the compositions. So we use tempo CNF and tragacanth gum with 30% of tragacanth and 70% tempo. And here we use 50-50. And after printing, those hydrogels were cross-linked with ionic cross-link with calcium chloride, which is very mild cross-linking. So we avoided to use any toxic cross-link or anything that we would need to make sure that the hydrogel doesn't have any contaminants or toxic effect on the cells. So here we have some rheology profile of these hydrogels. So here the viscosity, we see the shear thinning behavior, which is essential for 3D printing. And also we see the higher content of tragacanth gum also improve the viscosity of these hydrogels, because tragacanth gum is a highly branched anionic polysaccharide. So there is the repulsion or the electrostatic repulsion between the branched fibers and the main chain, and also the cellulose nanofibers, which improve the viscosity of this hydrogel. And if we look at moduli, the storage and loss moduli against shear rate, and here we have also the tempo CNF, which is the control. We have also the enhancement of the moduli with increase of the gum content in it. And also we have the elongation of this linear viscoelastic region, which is also essential for printing, and it should ensure that the structure stays with shape after printing. So we have the elongation of this zone. What we can also see in this graph is for the tempo CNF, we see overshooting of the loss modulus, which means that the fibers are interacting with themselves and resisting the flow. So you have this increase in the modulus in this part until it flows like a liquid. What we see is an increase in gum content, decrease this overshooting, which means that the fibers are being the tempo, tragacanth gum is absorbing onto the cellulose fibers and making like a lubricating effect, making it extrude easier in this sense. So after printing these scaffolds and cross-linking it with calcium chloride, we see here the swelling capacity of the tempo CNF and with 30% of tragacanth gum. And when we increase 20% more to 50-50, it enhances significantly the swelling capacity, mostly due to the electrostatic repulsion and the interactions with water, which absorbs more water. Which is this swelling is correlated to the porosity, which also enhances the cell vascularization and the nutrient diffusion inside the scaffold, the hydrogel. Also checking the compression properties of these hydrogels, we see the same effect. The increase in the gum content make the structure more stiff. So you have a higher compressions modulus, Young modulus. And if we compare with actually living tissues, it's in between skin and fat tissue. And we could also reach muscle and heart tissue. So you can modulate these properties to achieve a target application just by combining the polysaccharides. We also did some hemocompatibility of these hydrogels, and they were very low or no hemolytic. So they are not interacting with blood cells. We could also be used in topical use or direct contact with blood, not interfering and not clogging it. Also testing the biocompatibility, but in this case, we use a different type of cells. Here we use HEP2 cells, which are liver cancer cells. So here we have actually optical density, but it's directly related to cell viability. So the control, they have more or less the same, and they have more or less the same viability, showing no signs of toxicity. But in day 5, you have a significant increase, and you could also say that they are compatible and they could be used as platform for cell culturing. But also we wanted to see what's happening inside, how are cells proliferating inside the scaffold. So we did some fluorescence microscopy of these scaffolds. And this is the stacking of these fluorescence images. So we can see in depth that the cells are proliferating and migrating within the scaffold. And we could see until 500 micrometers the cells in the scaffold. So the fibers are not directly interacting with the cells, and the cells are being able to migrate and are so this system is a good environment for growing cells. So another example here, we use nanocellulose crystals, so CNC, and another polysaccharide, so xanthan, which is mainly composed of glucose, mannose, and glucuronic acid. Also a branched polysaccharide. So we mix in different concentrations with no hard cross-linking again to stay as natural as possible. So here we see the moduli against shear stress. So we have, it's a bit small, but I think you can see. We have the same effect, so only CNC here in gray. And when you increase the concentration of gum in it, so this is 70% gum, you have enhancement of the moduli and also enhancement of this linear viscoelastic region. And you can also see the decreasing of this overshooting of the loss modular. So we also printed those hydrogels, and actually it was very nice. This one has a very good definition. So we could also print in higher scaffolds or higher constructs. So this one has 4.5 millimeters with 9 layers of printing. And this one has 30 layers and reached like 15, more or less 15 millimeters. So here we have these two printing scaffolds, printed constructs, and we have this cross section of these hydrogels. So here we can see that it's very porous structure. Apparently you don't see any difference between them much. But if you check on the porosity and the pore size here, for the lower amount of CNC, we have 88% of porosity and the pore size of 84 micrometers. And when we increase the concentration of CNC, it hinders it becomes a bit more denser and compact structure, also due to the nature of the cellulose crystals, because they are more rigid compared to the flexible tempo cellulose nanofibers, which are longer and more flexible. So the increase in the CNC content diminished the porosity and also the pore size of the scaffolds. But if we check on the image, it's pretty, it has a really high resolution, you can see, and the surface is very smooth. And this size of the filament is 570 micrometers. So really good resolution of these CNC hydrogels. So also we did the compression tests, and we see that increased CNC content enhanced significantly the range and the compression modulus of these hydrogels. So here in the tempo CNF and here the lower concentration of CNC, we have these hydrogels in the class of skin and fat tissues. But when you increase 20% the concentration of CNC, it enhances to almost 20 kilopascals, which could be used as muscle and heart tissue. And also, if we check the viability of these hydrogels, we see that on day 1, they are more or less the same, but if we check on day 7, the control is pretty high, and the lower concentration of CNC had also more or less the same range of proliferation. But if we increase the concentration of CNC, we have a decrease in the proliferation of the cells, which is also correlated to the compression modulus. If you have more stiff hydrogel, that means that it hinders the mobility and the proliferation or the migration of the cells inside the scaffold, which is also correlated to the size of the pores inside the scaffolds. Was pretty low, was around 30 to 38 micrometers. So by just tuning and combining different polymers with different polysaccharides, we can improve and tune the properties to a target application, which is pretty nice. Only with polysaccharides, without any cross-linking. So the last part of this presentation, actually,
you have to conclude, Roberta. Thank you.
yeah. Sorry. Yeah, I'll be fast. Yeah, so this part, we were trying to understand the interactions between the cell and biomaterial with actual materials available and used as cell platforms for culture. So here we have cellulose and matrigel, which is a mix of polysaccharides and it's animal based, which is widely used in cell culture. So the aim was to understand how the cells proliferate and how they attach to each other to start forming the tissue inside these matrices. So we grow the cells in those two matrices for 1 and 3 days, and after 3 days, they start to attach to each other and forming these hydrogels. And we check the microscopy images and the gene expression. And we use 2 different cell lines for this, so liver cancer cells and some stem cells. So here we see the cells in cellulose. They grew on this spheroid, the cellular spheroids, very compact, while the cells, when grow in this matrigel, in these proteins, they grew scattered. So yeah, here we can see also the fluorescence images. Even in matrigel, we see some dead cells within these agglomerations, and which doesn't happen here in cellulose, we see only outside. So we wanted to understand how or understand the force between the cells. So we use 2 different techniques, colloidal probe microscopy and single cell force spectroscopy, to measure direct interactions between the cells. And here the cells coated with the matrigel and also coated with cellulose. So from this, we measure the deflection of the cantilever and we measure the detachment of the material or the cell to the cells on the surface. And we have this kind of pattern here for the force curves. So here are the force curves that we obtained in these experiments for the 2 cell lines. And so from the minimum part of the curve, we can extract the adhesion energy, the adhesion force. And for the area under the curve, we can actually find the adhesion energy between the cells and the materials. So here we could see some trends in this. We see that for HEPG2 cells, the HEPG2 interact strongly with other cells, then to the cellulose, then to matrigel. And for the matrigel, for the stem cells, the matrigel had a strong interaction with the cells, then the cells, and the cells in the CNF. But as you can see here, there is a very high variation of these values, so there is no significance difference between the matrigel and the CNF, and the cells are a combination, it's a long range of forces, of bonds. So we wanted to understand the breakage of every single bond interacting with the cell. So here we have the events, the breakage events per force curve. So we have these patterns. So this distribution of forces. And we combine these results with gene expression to understand what kind of interactions was happening. So in HEP2 cells, we see these scattering interactions also for these stem cells, but for HEP2 we have two types of integrins, of cadherins. So here we have these cell adhesion proteins in the cell membrane interacting with the other cell adhesion membranes, this type of interactions. While for Matrigel, what we saw was these specific interactions also between the integrins of the cells and the protein laminin composed of Matrigel, which is also a protein interaction. And for cellulose, we didn't see anything. We see only hydrogen bonds between the cell membrane and the cellulose structure. So comparing the two cell culture platforms, the Matrigel is widely used, but it downregulates the cell-cell interactions, and it relies on cell-specific interactions with the Matrigel, and the cells stay scattered because they prefer to stay attached to the matrix instead of attaching to each other, basically. And for the cellulose, it promoted the cell-cell adhesion because it didn't have any specific interactions with the cells, and provided the appropriate conditions for the cells to form spheroids and start forming tissue. So concluding, these hybrid polysaccharides, they can be combined and to tune the properties. So only combine the properties of this, you can enhance the mechanical properties and the rheological profile, and you can have controlled over porosity and pore size. And these materials have good hemocompatibility and biocompatibility. Also, they can be used as efficient drug release platforms, and they promoted cell-cell adhesion. So, yeah, thank you. I would like to thank a lot of people that help and were participating in all these projects. And thank you all for listening, and Professor Pedro for giving me the opportunity to speak a bit about my research. Thank you.